Welcome. You are about to view a recorded talk of the Progress and Visions in Consciousness Science online seminar series. This series is organised jointly by the Association for Mathematical Consciousness Science, AMCS, the Mediterranean Society for Consciousness Science, and the Oxford Mathematics of Consciousness Network. While every session consists of a talk and discussions, the latter are not recorded and the following will only contain the talk. We hope you will enjoy it. Further information and details on how to join are available on our website.
So, well, welcome everyone to the first talk of our seminar series, Progress and Visions in Consciousness Science. So this series has been in the making for quite some time, because there were ideas that people would want to have a series again. And we just had wonderful experiences in the past, both in terms of content with previous series, but also in terms of just to get to know people, online, and then you would meet them at conferences, and many wonderful experiences grew out of that. And so I'm very glad that we do that again. And I think we've given it enough time and enough distance since this very intense online period, which we all had during the pandemic. And so hopefully everyone is enjoying a good online talk again. And yeah, it's our great pleasure to have a very, very renowned speaker for the very start, which my colleague Andrea is introducing just in a minute. So what I wanted to add just before we go to the introduction is just a little bit of information about the structure of the session. So the talk will last approximately 45 minutes, and we would kindly ask everyone to stay muted during the talk. You're cordially invited to share your webcam. Tradition is, if you share your webcam, we'll greet you personally, just because it's a bit difficult to keep track of the participant list. But you can share your webcam or not, whatever is comfortable for you. And then after the talk, we'll go over to a question and answer session. And if you wanted to ask a question, we kindly ask you to just raise your hand in Zoom. You can use the reaction symbols here. If you click on reactions, there's a raise your hand symbol, I think. No, I activated some new, we find it later. Anyway, and then we will call you up, and you're cordially invited to then unmute yourself. And also, if you feel comfortable, you could share your webcam feed to make it a little bit more personal. You don't have to, though, if you don't want to. But it would be great if you just can ask your question personally as compared to the chat. And then we have, I think the original goal was to have 15 minutes for question and answers, and we'll see how that goes. Maybe if we need a little bit longer, we'll kindly ask Kathryn how she's doing on time, but we'll try to aim it at that somehow. Okay, so big thank you again for being here, and I hand over to Andrea, who's the pilot and mind behind today's talk. Thanks very much, Andrea.
Thanks very much to you for you to be here. I'm very happy and excited to welcome you in the first session of the seminar series of this year's entitled Progress and Visions in Consciousness Science. It's my pleasure to introduce Professor Hess. She's an algebraic topologist in the broadest possible sense, a full professor at the EPFL Lausanne, and her research in pure mathematics covers topics like homotopy theory, higher category theory, and algebraic K-theory. But not only that, she has been a pioneer in the applications of algebraic topology in the special sciences, with applications ranging from knot theory in polymer science to other tools from algebraic topology in neuroscience. And I'm very happy that we are here today to hear her about this topic. She's also received many awards for her teaching and research abilities. Notably, she is this year's chair de la Vallée Poussin in UC Leuven. So welcome you, and thanks, Kathryn, again for giving us this opportunity.
Thank you very much for the very kind introduction, Andrea. It's a great pleasure to be here. I'm not going to be saying much about consciousness, I'm afraid, because that's not really one thing that I've worked on. But I will be talking about applications of algebraic topology in neuroscience more generally. And then my colleague and collaborator, Lida Canary, will be with you next week. And she'll be talking about how some of the tools I'll describe today have actually been used for more in-depth studies of how neurons, the neuron morphologies, how neurons connect based on their morphologies and things like that, and maybe get a little bit closer to consciousness. So today I'm going to talk about how we can use topology really to analyze networks of neurons. So that's our goal today. And so let's see, just get there we go. So I don't expect everybody in the audience to know what algebraic topology is. So I'm going to say a few words about that, and then say some words about how we use algebraic topology to analyze data in general, and talk about an application of that related to neuroscience, and then get more specifically into how we can analyze networks of neurons or networks of brain regions or networks in general, in fact, but we'll be focusing on networks of neurons. So the cool thing about algebraic topology, it gives us a way to describe the structure and function of networks using a common language. And that's what I want to sort of highlight today. So we're going to start by talking about topology and data science. How could we use something like algebraic topology, which is this area of mathematics, which you can think of as being sort of somewhere between algebra and geometry, to study something like data science, which seems very different, very distinct from an area of pure mathematics like algebraic topology. So what is topology? So to start with, we could say it's just the mathematics of shape. And so even if you're not a mathematician or not an algebraic topologist, you probably maybe have even heard of this joke about how algebraic topologists can't tell the difference between a coffee cup and a donut, because algebraic topologists are thinking about shapes up to continuous deformation. And so this little video, which is running now, is the illustration of converting a donut into a coffee cup, where you're thinking about these objects as being made of some sort of extremely extensible material, like an infinitely extensible rubber or something like that. You're allowed to compress, to stretch, but you're not allowed to cut or to paste things. And under those conditions, we could say that two objects were equivalent in the eyes of a topologist. And it's also the mathematics of connectivity, which is it's really particularly good. It provides tools for understanding connectivity. So here we have some sort of connected network, and we're going to come back to this idea later. And so it's not just telling about sort of which nodes in this network are connected, but how are they connected, and how many different ways, what are the relationships among the ways that they're connected, and so on. So topology is really great for providing you with tools to talk about things like that. But perhaps even more importantly and more interestingly, is that it's one of the flavors of mathematics that's really good for describing the emergence of global structure from local constraints. So this is something that we're often very interested in from various perspectives, that locally, sort of within a little neighborhood, what things look like, but then there are rules about how you can glue those local pieces together that can tell you about how you have the emergence of a whole global structure. And so this is relevant in the context of neuroscience, for example, because we have a lot of local data about how different kinds of neurons connect, or like what happens in little tiny regions and things like that. And you want to see sort of how does the whole global structure emerge from that, and properties of the global structure emerging from these local constraints. And so the example of this is the Möbius strip, where locally it just looks like a little interval across another interval, but the way that it's all glued together gives you this twist in the Möbius strip. So that's one kind of mathematics that can be really good for answering these kind of questions. So what in the world does this have to do with data science? And so we're all all over biology, psychology, all over natural sciences in general. There's a huge amount of data being created. And topology provides us with interesting tools to provide a new perspective on how we can understand that data. So the idea is the following, that it's really the shape of a data set in some way that we can characterize using algebraic topology, that we try to find some way of describing that shape by some kind of topological signature, whatever that can be, which would be, as we'll see examples of this, it can be just numbers, or it can be a curve, or it can be something more complex, but still simpler to understand than an entire data set, which can encode the structure of this data set sort of along a range of scales, because often when you're analyzing data, a lot of the standard tools require you to choose a threshold. So you're choosing a particular scale on which you're understanding the data. But the nice thing about the way we're going to apply these topological tools is it allows us to consider multi-scale processes, and you can sort of and which can lead you to understand better what are the important features in your data set. And so this was this idea has led to the development of the field. It's called topological data analysis. And I'm going to give you a little hint of what we can do with topological data analysis now. So what kind of topological analytical tools do we have? So here's a list of some tools. One of the earliest tools that was developed it's called Mapper, developed by Gunnar Carlson and others, that is used really, which is really good for studying various kinds of clinical data, metabolic data, genomic data, and so on. There are versions of it that are such as two-tier mapper, which is particularly good when you have smaller data sets that can, with single cell data, for example. There's persistent homology, which I'm going to spend more time on, and then you can think about graph signal processing. So there's a whole range of very interesting topological analytical tools that have been developed. More recently, people have looked into integrating some of topological notions more closely into machine learning tools and so on. But what we're going to focus on today is what's called persistent homology, which is really good for analyzing connectivity data in large networks, or considering very high dimensional point cloud data. So when you have your data that are, for example, with gene expression data, when you have thousands of components that you're looking at, and so there's some sort of high dimensional data that you're studying. The basic workflow for persistence goes as follows. You have whatever your data is, you want to try to convert it into some sort of point cloud. So this is some sort of embedding of your data into often a high dimensional Euclidean space. In general, you want to embed it into some sort of metric space so that you have a notion of distance, or at least of similarity between your points. From that, you're going to construct some sort of a filtered geometric object. So that means you're going to look at some sort of sequence of geometric objects, one included in the other. So a very toy example would be, say, okay, I have half an annulus that I include into an annulus, and then there's another cross piece that introduced, and then I fill it all in. So you have this sort of sequence of geometric objects where one is always included in the other. We call this a filtered space, and we'll see examples of this in just a moment. And the idea is that you're going to take this sort of filtered data, which is coming from the point cloud, and it's going to spit out some sort of topological signature, one of which is called a barcode, and which is why I've used an actual barcode here, as a way of encoding this data, very complex data, in some sort of more simple form that is easier to introduce into standard machine learning tools, for example. So the key notion here is really this notion of having a filtered object that you're studying. So let's see an example of how this could work. So how do we get from a data set to a point cloud, for example? So one example would be, suppose you have some sort of time series data. So you have some sort of fluctuation over time. So time would be along the horizontal axis here. And then there's something you're measuring, which is exhibiting this kind of periodic behavior. And one standard tool for analyzing data like this and converting it into a point cloud, that is to say some bunch of vectors in a Euclidean space, is to say I'm going to do a tokens embedding. I'm going to choose fix some number of points I want to think about, fix some distance in time between these measurements. And also then I'm going to take this number of points, in this case be 3, and then shift it a certain amount over time. And it's going to give me a sequence of vectors in R3. So it gives me a sequence of points in R3, and you're converting this sort of periodic behavior of the curve into something that actually you see, now you see this sort of cycle behavior, this sort of circle behavior, by doing this tokens embedding of this time series data into R3. So this is very often applied to time series data, and it's become quite standard to then apply the tools of topological data analysis to point clouds that are coming from this kind of tokens embedding. So what do you do once you have a point cloud like this, so a bunch of points in a Euclidean space, a metric space at least, where you have some notion of distance? Well, one way to do this is to do what's called either a Vittorius-Rips or a check complex, which is to say around each point, you're going to consider a sequence of balls of increasing radii. And when the balls centered around 2 points in your data set intersect, then you're going to include an edge. And when you have three different balls that intersect, and you have all three edges, and then you fill in the triangle as well, and so on. This is a way you can build up gradually a larger and larger space. So here we're looking at a radius of 0.3 around these points. And then we slowly, as the radii grow, you include more and more edges like this. And the radius grows more. And now we see, for example, here and here, that we have also triplets of points that are connected by edges. So we would be filling in the triangle, and here we would fill in a tetrahedron, and so on. So we're building up these more and more complex geometric structures, and the balls get bigger and bigger, and we include more and more edges and more triangles and tetrahedra and so on. And so we have these more and more complex geometric shapes until once we've reached a certain radius, and all these balls are intersecting. And so we have this sort of a natural stopping point for this filtration. So this is how we can go from a point cloud to a nested sequence of complexes like this. Once we've done that, then how do we get this sort of barcode or some other topological signature out of it? So here's an example. So we're going to look now, maybe I'll even get a pointer here. There we go, there's a pointer. So here we have a sequence of 4 complexes like this. So we have always so we even, in this case, we're even adding sometimes new points to the collection, but there's always, each one of these is included in the next one. And what we can do is sort of distinguish particular topological features, particular important structural features of this data that we want to measure. For example, we could say, how many different components do we have? So connected components. So here we would have 3, here we have 4, here we have 2, and so on. And that's usually denoted beta 0. Beta 0, beta 0 here is measuring the number of components. So the number of components is 3, 4, 2, and 2. So that's the sort of information you might like to retain about this sequence of complexes. You might also say, okay, no, actually I'd like to count how many loops do I have? How many empty cycles do I have? So here there are no loops, there are no cycles in here. Here I have a cycle. So here I have 2 of them, because this one now is filled in with a triangle, and here I have just 1. And so I could say, okay, what I'm going to call beta 1 or beta 1 is counting the number of loops that I have. So 0, 1, 2, and 1. And then I'm going to record this data in the form of what we now actually call barcodes. So it's keeping track of the number of, for example, components as the filtration step increases. So we have, for example, we have these 3 different components, and some of them continue forever, and others are sort of glommed into other ones. So for example, here we have one component that's starting here, but then because we add these 2 edges, it becomes included into this one. So this is a component that's born in step 1, and then it dies in step 2 because it becomes part of another component. And for example, similarly, here, this component is born at this point, but then it dies here because it becomes part of this one. So you sort of follow when does a component appear, and then when does it become part of another one? And that's what's measured by this barcode. Similarly, when we're looking at dimension 1, when we're looking at loops, here we have a loop that is born at this stage, but then it's filled in at this stage. So that just gives you a bar from 2 to 3. Or we have a loop here that's born at stage 3, but then it's filled in at stage 4. So that one just gives me a bar from 3 to 4. This one is born at this stage, but then it continues forever, and it goes like this. So we have this way of recording what is sort of important topological features as we move through this filtration, and that's what this data is recording. And it seems to be like a huge dimension reduction in terms of information, but it is actually, it is a helpful way of keeping track of important structure, and to see how that structure sort of persists. That's where the persistence and persistence homology is coming from as you go through the filtration. So these, for example, would be features that really persisted, and these are sort of more ephemeral. They come and they go. Okay, so what do you do with barcodes? I just want to point out that we can also, there's an equivalent way of approaching barcodes in terms of what are called persistence diagrams. So here I have a barcode. I have a bunch of these intervals. A barcode is nothing but a set or even a multi-set, because you can have multiple copies of the same interval. But so it's a bunch of intervals like this that are telling you when features appear and when they sort of get agglomerated or they into other features or they disappear. So we have this bunch of intervals, and anytime you have a bunch of intervals, what an interval is always determined by what its endpoints are. So you could simply record the endpoints instead. So here I have an interval from 0 to 2. I can record it as a point in the upper right-hand quadrant as the point 0, 2. Or I have this one here, which is going from, what seems to be 1.5, 3.5 to 3. So this would be correspond to this one here. So each interval here corresponds to a point in this persistence diagram, and people go back and forth in terms of how they represent this topological information, either by the barcode, which is very visual, or by the persistence diagram, which is something a little more, which lends itself perhaps better to thinking about a space of persistence diagrams or various generalizations thereof. So these are the kinds of topological signatures that are often used when people are trying to encode interesting shape data about shape information about data sets. Okay, so here's some sort of more technical information. If we look at the set of all barcodes or all persistence diagrams, there are various ways you can put distances on this set and make it into metric space. So there are various earth mover type distances, one of which is the Wasserstein distances, which are of LP type, or the bottleneck distance, which is of L infinity type. But I'm not going to go into that kind of technical detail today. And when we think about this whole instantiation of the pipeline, we would like, when we get these topological signatures, we would like them to be stable in the sense that if I have two data sets which are not very different, then the output at the end of this pipeline, the two different barcodes that I would get, shouldn't be too different. And so that's what, when I say that this is Lipschitz continuous, that means that if we have 2 point clouds that are similar, close to each other in a certain sense, in the sense that the Hausdorff distance between them is small, then at the end of the pipeline, you end up with two barcodes that are actually close to each other with respect to one of these earth mover type distances here. So we have stability guarantees, which are really important if you want to be able to interpret the output of this pipeline and be able to use it to be able to make any conclusions about the data that you started with. And if you're interested in exploring these kind of tools, there are fantastic software libraries that are available now. Almost all of them are open source for doing all kinds of TDA computations and also integrating them into various machine learning pipelines as well. So I just mentioned, I highlighted Joto TDA because it was developed by an EPFL startup, a bunch of EPFL students that created a startup that now has a nice TDA software. And there's even a version called Joto Deep that integrates deep learning with TDA for then, taking these topological signatures and actually using them for doing various kinds of machine learning analysis. And then in terms of, once you have the output, you have these barcodes or whatever topological signature you're using, you'd like to be able to make conclusions about your original data set. So you have to have some kind of ways of doing inverse analysis and saying, okay, if I have a barcode like this, what can I deduce about the original data set that I have? What features am I picking up? What is important? How does this tell me where the important areas are in my data set? And so there are various inverse analysis tools that are available. And I think Lida may talk about something like this next week as well. So I just want to give one example of an application of this pipeline that was used in something related to neuroscience, very far from creatures that are very conscious, perhaps, because we're talking about fruit flies and even larval fruit flies. So these are incredibly small little animals. But my colleagues here at EPFL, who are very, very good at using highly sophisticated machinery to study these sorts of creatures, and they could actually use what's called light sheet microscopy, which is what's illustrated here, to study the brain of these larval fruit flies. So these brains have roughly 8,000 neurons, something like that. So they're quite small, still bigger than C. elegans, but still quite small. And so they did analysis, and they could actually find the precise 3D coordinates of all of the stomata, so all of the cell bodies of the neurons in the brains of these larval fruit flies. And that, okay, well, this gives us something that is literally a point cloud, right? We have 3D coordinates for a bunch of points. And can we say anything interesting about this point cloud using topological data analysis? So the first, they had just 4 or 5 of them, and the postdoc with whom I was working on this, Gard Spreeman, he said, yeah, well, we don't know, there's nothing to compare it to. We just have, there was nothing. But once they actually got, I think they had 50 or 100 of these brains that were done, started looking at it closely and realized that actually, very surprisingly, there's a sexual dichotomy in the brains of these larval fruit flies, so between males and females. So if you look at a fruit fly larva, you cannot tell the difference from the outside about whether they're male or female, but their brains are already quite different, apparently. So what I'm showing you here, we took these point clouds and got the associated topological signatures, the barcodes, and tried to use the notion of distance between barcodes using machine learning in order to distinguish, to see where we could distinguish between males and females. And what this plot is showing is that if you do a control versus having the actual data, you can actually distinguish quite well between the males and females using this topological data. Whereas on the right-hand side, this was just saying, let's just look at the numbers of neurons rather than their actual locations, not using this sort of geometric or topological information that's coming from the whole point cloud. And then we really couldn't tell the difference between the males and the females. So this is, they did not expect there to be a difference, and it was something that came out only because we looked at it with the tools of topological data analysis. Kind of cool. To move on to networks of neurons. So just a little reminder for those who are not actually neuroscientists in the audience, which I have no idea whether that's how many people actually know much about the neuroscience. I know a bit about neuroscience. I'm not actually a neuroscientist. I'm really an algebraic topologist, but I hang out a lot with neuroscientists. So just to give a little bit of neurobiology. So when we're looking at a neuron, it has 3 important parts that we need to distinguish. So they're the dendrites, which are these sort of finger-like structures reaching out here that are gathering information from all around the network. And then there's the cell body itself. And sort of as electrical signals are gathered by the dendrites, and there's some processing already of the information that's going on in the dendrites, what happens is that the electrical potential on the membrane of the neuron rises. And when it hits a certain threshold, it may or may not emit what's called an action potential, or a spike, that would be transmitted along the axon here towards the dendrites of another neuron, and where the signal passes from the axon to the dendrites via what are called synapses. So these are the connection points between the axon and the dendrite. There's an illustration here of the electrophysiology of how the signal is transmitted and so on. We're not going to talk about that today, but just to say that the important information idea I want to transmit here is that there really is this idea of the flow of information along the axon to the dendrites, and then they have these connection points at the synapses. So here we have illustrations coming from the Blue Brain project, a beautiful visualization where we see the cell bodies of two neurons, and you see that they're actually considerably more complex than they were in this cartoon that I was just showing you. And the idea is that we're looking at these highlighted points here should correspond to the synapses that are the connection point from the axon to the dendrite. One sort of side note to make here is that in order to have a reliable connection between 2 neurons, they actually have to share several synapses, because synapses are also have a stochastic sort of behavior. They're not going to transmit the signal every time via their neurotransmitters. So, but the main point here is that these connections are connections with a sense of direction. We need to take this sense of direction into account. And so how are we going to do that? We need to think about what sort of mathematical representation we want to make of a network of neurons. And the idea here is, because we have this natural sense of a flow of the signal along the axon to the dendrite, that we're going to represent the whole circuit of neurons by a directed graph or a digraph. And because we cannot take all of the information that we have at our disposal not even all of it is at our disposal, but all the information about the network of neurons be too complex in order to analyze. We vastly simplify the representation we're considering and just say, okay, let's just think of it as a directed graph. A directed graph in which the nodes of the graph are going to correspond to the neurons, and the directed edges are going to correspond to a connection from one neuron to another via some number of synapses. We're not even going to think about how many synapses are involved. Let's say there are at least some synapses that are giving us a connection from this neuron to this neuron. So this is, a very small version of such a thing, because if you're thinking about an actual network of neurons, for example, in the brain of a rat that has hundreds of millions of neurons, the brain of a human, hundreds of billions, here we're just thinking about a very small number. But even so, we can see, well, how could we characterize or describe such a graph? How do we extract what's important about such a directed graph? And one more point I want to make about this is that as in a brain, we can have reciprocal connections. We can have an edge going from this neuron to that neuron, and we can have an edge going back. There's always a fairly sizable fraction of neuron pairs that are connected in both directions. So how can we really appropriately characterize and describe the structure of a complex directed graph? I'm going to give you one approach that uses the tools of algebraic topology to do so. So the idea is, and this can be applied to other kinds of networks as well, is you say, okay, well, what is really important about this network? So what would be sort of the significant pieces of this network? So I'm calling them significant subnetworks. And you want to find something that's actually going to be appropriate for the network that you're thinking about. In our case, we're thinking about a network of neurons, which are really going to have directed connections. And then we say, okay, well, what would be significant subnetworks in this case? Once you've made that decision, which is based on your context, then you can give yourself sort of local information about the network by doing a very basic statistical analysis where you say, okay, how many times is a particular kind of subnetwork represented within this really big network that I'm thinking about? The idea is you're going to focus on pieces that are actually small enough that they're sort of understandable, and you want to say, okay, well, how are they pieces like that distributed throughout the big network? And so by just doing that kind of counting, it's giving you sort of local information about the network. But you could ask, well, how do these pieces actually fit together? Because you could have multiple copies of the same kind of subnetwork within the bigger network, but how do they overlap? How do they fit together? How do they together build bigger and more complex structures? And this is, again, this sort of emergence of global structure from local structure. And so that's when you can find some way of quantifying that, and you're giving yourself more global information about the network. So in the case we're thinking about directed graphs, an interesting collection of significant subnetworks to consider are those we call directed n simplices. So if we just use mathematical language, we'll say it's going to be any completely cyclic subgraph on n+1 vertices. But let's see what that actually means. So a directed zero simplex is just a vertex, just a node of the graph. A directed one simplex is just a directed edge. It's an acyclic subgraph on two vertices. A directed two simplex, okay, now we have three nodes, and being complete means that every pair of nodes is connected, and being acyclic, it means there are no cycles. So that means that there's always this sense of direction, a unique sense of direction like this. So we are going to have a source node, a sink node, and at every node we know this, okay, where we're going with this well-oriented sense of direction. The idea is that we're looking at the feed forward pieces of the network. And then we can push this even further. This would be a directed three simplex with four nodes. And we notice we have a source node, we have a sink node like this. And so we'll just use this sort of terminology for these, or this notation for these directed n simplices. And so we can continue this, obviously, for any n in this process. And the idea is we just have these feed forward pieces, which are sort of more and more robust ways of transmitting information from the source to the sink. robust in the sense that here, for example, if I lose this one edge, that's it, we're done. We're not going to get information from here to here. In this case, if we lose one of the edges, we still have a way to get from the source to the sink. If we lose this one, or if we lose this one, or if we lose this one, we still can get the information there. And the more connections we have like this, the more robust it is. It's a feed-forward process. So that would, if we wanted to get sort of local information about the directed graph, we could just simply count how many of these directed instances there were. But if we'd like to sort of figure out how the pieces fit together, this is where the notion of a directed flag complex comes in. So this is a generalization of the notion of flag complex or click complex. And in order to understand this picture, we just notice that if we have a directed in simplex, so in this case, let's take a directed to simplex, if we remove any one of the nodes, then what we're left with is a directed edge, in other words, a directed one simplex. And this is true for any n. So in particular, in this case, I could remove this node and I have this edge left, or I can remove this node and I have this edge left, or I can remove the bottom node and have this edge left. And so similarly, you can see that anytime I have a directed in simplex, if I remove one of the nodes and all of the associated edges, I'm going to end up with an n-1 simplex. And there are n+1 ways to do that because you have n+1 nodes. And so this is sort of giving us a way of understanding how the n simplices for different n can sort of be fit together as building blocks of bigger structures within a very large graph. So what we'll call the directed flag complex is just the collection of all the directed n simplices in G for all n. And it's what's called an ordered simplicial complex, which means simply that it's a collection of lists, which is closed under taking sublists. So if I have lists, I can take any sublist and it's still in there. And that's this business of being able to remove nodes and still having a simplex. So the flag complex of G is just a set of all subgraphs that are isomorphic to, that are a copy of one of these directed n simplices for some n. And so an example of this would say, okay, here's a very small directed graph that even has a nice little reciprocal connection here. And we're looking for the directed simplices. So if I build, so here's my G, and I build my flag complex, then here, for example, I have a directed to simplex because it doesn't have any cycles. So I'm going to fill that in with a triangle. This one here, this is a cycle. So this is not going to give me a directed to simplex. If I go like this, here's another directed to simplex. So I fill that in with a triangle. However, if I look at this reciprocal connection here, it's giving me a cycle again. And so that's not going to give me a directed to simplex. And so what I'm seeing is I get this sort of geometric structure by filling in with triangles where I have directed two simplices. I'd fill in with tetrahedra, I have a directed three simplex, and so on. But what's interesting is the cycles are still important because they're creating sort of cavities. They're creating gaps or spaces in this structure. And we're going to see how we can formalize that. The idea of a cavity, if we want to call it that, or a homology class, if we'd rather use more fancy language, is that we're building up something that looks like a loop. So here's one that's built from 4 one simplices, or in a more complex way, we can start with some two simplices, 8 of them. And by gluing these two simplices along their faces, along their edges, we can build a triangulation of the sphere. So it's this a cavity, there's a three-dimensional cavity that's enclosed by this. So we call, and this is going to give us, call it 2 cavity. We can make this from 8 of these two simplices. Other examples, here's another example of a loop, or we can build things like tori and such. Okay. So from the flag complex, we can get various global numerical descriptors of our directed graph. So one of which it turns out to be incredibly useful, is to say, okay, I'm going to look at, for each k, I'm going to look at the number of k simplices. I call this little fk of g, and that are in the graph. And then I just look at the alternating sum of these numbers. So the number of vertices minus the number of edges, plus the number of directed to simplices, and so on. And then with this sort of alternating sum, so if n is the highest dimension of simplex you find, then it's plus minus 1 to the n times the number of n simplices. And this turns out to be, it seems to condense a huge amount of information of the graph just to one number, but it turns out to be a remarkably useful global numerical descriptor of your graph. Another would be what we call the case-directed Betti numbers. So these betas that I mentioned earlier are coming back, Betti numbers, which is counting the number of k-dimensional cavities in your structure. So the number of, if k is 1, you're looking at the number of loops. If k is 2, then you're looking at sort of the number of sphere-like structures in your network, and so on. So this gives you, and this one is really a very global descriptor, because again, you're looking at how you can take these local pieces and build more complex structures from them. So here's the example again. So in this case, we have 6 vertices. So F0 of G is 6. We have 12 edges, directed edges. So F1 of G is 12. And we have 8 faces, 2-dimensional 2 simplices. So that gives us 8. And if we count these, look at these Betti numbers. So the 0th Betti number is again looking at the number of components. We have 1. The first Betti number is looking at the number of loops. There are no loops, so that's 0. And Betti 2 is looking at the number of 2-dimensional structures you have like this, and there's 1 of those. And what's kind of cool is that you look at the Euler characteristic, which is the alternating sum of these 3, 6 minus 12 plus 8. It's always equal to the alternating sum of the Betti numbers as well. So these 2 sets of numbers, which seems to be quite different, actually, when you take their alternating sum, gives you the same thing, the Euler characteristic. So this is the kind of information that we can extract from a network, from a directed graph. So we applied this in an article that appeared in 2017 to the blue brain reconstruction of part of the brain of a rat. So this was done by the Blue Brain Project, which is affiliated with EPFL. And they did a digital reconstruction of part of the brain of a 14-day-old rat. So this is on a computer, taking into account all sorts of biological data. And the algorithm that was used to do this reconstruction has various stochastic elements, and so to have some sort of variance among the different models to come up, they actually, for the same sets of biological input parameters, they did 7 different reconstructions, instantiations. So they had 42 different circuits they built like this. And it was not just the architecture was important, they could also simulate activity in this network, both spontaneous activity, neurons just firing just because, and evoked activity when neurons are firing because there's actually some input stimulus that's given to it. So we took this network and we said, okay, what can we say about it? Is there something special about it in terms of complexity? And so we looked at the numbers of simplices of various dimensions that there were. So in this figure here, the blue curve here is looking at the numbers of directed simplices of various dimensions that we have in this blue brain network. And so the blue brain network has roughly 30,000 or 32,000 vertices and roughly 8 million edges. So it's already a very big directed graph. And I wanted to see, okay, what sort of directed simplices show up? And what you see is that if there are quite a lot. There are 80 million 2 simplices, roughly 65 million 3 simplices, and so on. There are even, as we see here in this little blowout, some 7 simplices. So we have 8 different neurons that are all connected in this interesting feed-forward way. You say, okay, those are just numbers. We need to compare them to something. So I compared it, for example, to an Erdős-Renyi type random graph, which has the same number of edges and the same connection probability. And so this is just saying, okay, I have 2 neurons and I have some probability connection would be the same as the average connection probability for the blue brain network. And you see that if you do things randomly, then you have many fewer of these simplices of much lower dimensions, and therefore that somehow the existence of these simplices is picking up some sort of non-random information. So doing a comparison with an Erdős-Renyi random graph is not really fair, because we didn't expect it to be random in that sense, but did a comparison with other more simplified biological models and see that in those simplified cases, even there, you don't get as many of the lower dimensional simplices, and the simplices, you don't get simplices that are as high dimension. So this is somehow giving us a way of quantifying the additional complexity of this biological reconstruction. And then looking in the lab, you saw that actually you do see these simplices in actual brain tissue. So this was coming from brain tissue of actual rats, in which they were just sampling up to 12 neurons to see whether they were all-to-all connected. And they even ended up with some of these 4 simplicities. So 5 neurons connected, 5 out of 12, which is really quite striking, connected in this acyclic all-to-all connected way. And then did in silico version, so an on-computer version of also doing sampling up to 12 neurons in the brain, in the blue brain reconstruction, seeing that it looks like a similar kind of distribution. And if anything, the blue brain reconstruction is probably underestimating the actual density of such feed-forward structures in actual brain tissue. And you could say, okay, fine, those are certain structures. Do they actually have some sort of functional role? And so that was the next question you looked at by looking at simulations of activity in the network and saying, okay, if I have 2 neurons that are connected, is there what is the correlation in their electrical behavior? So you look at the sequence of action potentials of spikes that they emit, and you do a correlation between these what are called spike trains, and to say, okay, what sort of correlation do we see in this electrical behavior? So what we see here at the line at roughly 0.3 is the average correlation in electrical behavior between 2 connected neurons that are connected throughout the entire network. But then what's called dimension here is the maximal dimension of a simplex to which a connection belongs. And what we see is that as the maximal dimension of a simplex to which a connection between 2 neurons belongs, as it increases, the correlation electrical behavior increases as well. And so this means that being part of an actual simplex like this increases the coherence in the electrical behavior of the neurons. And the different colors here are just talking about where in the simplex you have it. So the red one would be sort of the connection between the last neuron in the simplex and the one just before it. Or purple would be from first to last and so on. And you see that, so the correlation increases as the dimension increases, but also it's a function of where in the simplex this connection lies. So there is something going on. There is a reason to focus on these directed simplices, these little feed-forward pieces, because they do have something to do with the coherence and the electrical behavior of the neurons. This is something that we later studied as well from the perspective of information theory that also seems to shed some interesting light. So this next figure is one that actually wasn't in the paper that we published because we didn't have the computational power or a correct algorithm to be able to do this computation before the article was published, but I think it's really important to talk about. So as I said, they were looking at input data actually from 5 different rats, and then there was some sort of average data. There was biological input that you could put into the reconstruction algorithm. And then for each of the sets of biological input data, there were 7 different instantiations or 7 different circuits that were built because, as I said, the reconstruction of these circuits has stochastic elements. And so even if you have exactly the same input data, you're not going to have exactly the same output structure. So you could ask, well, if I have 2 circuits built from the same input data, how similar are they topologically? Or if I have 2 circuits that are coming from different input data, are they similar? Are they different? And so on. And so here we were plotting what algebraic topologists called Betty 2 versus Betty 3, the number of 2-dimensional cavities versus the number of 3-dimensional cavities in these networks for these different input criteria. So these are different colors, and then light blue is the average. And you see that it's incredible, in my perspective, how well you can distinguish between the different types of rats, so the different input data, based on this topological data. So topologically, the circuits that are coming from the same input data are close to each other. And if I've circuits coming from different data, you end up with topologically different circuits. So I thought this was really, really fascinating and shows that topology is actually picking up something interesting biologically as well, because the only difference here is the difference in this biological input data. Also validated this sort of analysis on C. elegans, so little nematode, on looking to see, okay, C. elegans has only 279 neurons, at least in this particular model, and then looked at the numbers of simplices and embedding numbers. This is a case where we know the entire network, what it looks like, and did the same sort of computation in this case. And you see that, you know, it has quite a lot of simplices for such a small number of actual neurons, and that it even has 7-dimensional cavities in this structure, which is something that's very complex to build. It's definitely not going to happen randomly. And did the comparison with Erdős-Renyi random graphs with exactly the same number of nodes and the same average connection probability. We see again that it's a much less complex network measured in this way. So recently, the Blue Brain has built a much bigger model of this. They've looked at the whole somatosensory cortex of the rat instead of just looking at one little column with 31,000 neurons. They're now up to 4.2 million neurons that they could fit together with 13.2 billion synapses. So this is an incredibly complex network that they're able to build for an entire part of the brain of a rat. And you could see that there are similar sort of patterns that show up. If you just look sort of at local connections, you see here, these are all the connections you see of, again, simplices up to dimension 7. You have the same kind of curve for the number of counts. The numbers are much bigger. This is the logarithmic scale here. And if you compare it to various random models, such as Erdős-Renyi's stochastic block model and so on, then the numbers are much smaller. And here, you're taking into account also not just the sort of local connections, but also long-range connections. And you see, if anything, that you get incredibly complex structures with simplices up to dimension 18. The counts are enormous. And again, if you do the comparison to various random models, then the numbers are much smaller. So you really are picking up something important and interesting about the complexity in this case. So I'll stop there. Thank you very much for your attention, and I'd be very happy to answer any questions.